Hello, everyone. My name is Elle, and I am pleased to be talking to you today about my research on neurodegenerative diseases, specifically Alzheimer's and chronic traumatic encephalopathy. I will be discussing my review research on the prominent molecular mechanisms of each disease, respectively, in order to decipher unifying characteristics for therapeutic targets. Before we start, I'll share a little bit about myself. I'm presenting from my home here in Orange County, California, where I'm a senior at the Orange County School of the Arts. Alongside my scientific interests, I study popular music, vocal performance, and multi-instrumentalism. My passion lies within neuroscience, which started as an interest in neuroplasticity and has since grown to molecular and cellular biology. This research topic ultimately allows me to delve into molecular neuroscience, prepare for future lab research, and deepen my understanding of neurodegenerative disease. To begin, we must discuss the significance of the normal functioning protein tau in neurons. Tau is an essential microtubule-associated protein predominantly expressed in nerve cells, where it plays a crucial role in promoting the assembly and stabilization of microtubules. Essentially, in neurons, microtubules form an integral part of the cell's cytoskeleton, influencing its shape and function. This allows for kinesis-mediated transportation of cargo in neurons, therefore making neurotransmission more efficient. Broadly speaking, in cases of pathology, abnormal alterations of tau lead to the development of neurodegenerative diseases known as tauopathies. This protein is ultimately encoded by the mapped gene on chromosome 17q21, which is crucial in tau-related neurodegenerative diseases due to various alterations in tau production, including isoforms and mutations. The key difference in map transcription is the production of 6 major isoforms via alternative mRNA splicing, particularly 3 or 4 microtubule repeats. The isoforms are listed here. And the balance of these isoforms is vital for normal neurotransmission, and any imbalance or abnormal expression ultimately contributes to selective vulnerability of neurons. However, following the transcription of the MAP gene, tau protein undergoes phosphorylation. In the case of neurodegenerative disease, tau proteins undergo abnormal phosphorylation, which is also known as hyperphosphorylation for different reasons, which ultimately causes tau to detach from microtubules and lead to the formation of insoluble aggregates. These aggregates are known as neurofibrillary tangles, or NFTs. NFTs are also known to spread in a prion-like manner from neuron to neuron, propagating tau pathology in neurodegenerative diseases. This diagram shows the dominant characteristics of Alzheimer's disease and chronic traumatic encephalopathy. While this may not be totally apparent in the diagram, these two diseases are very distinct, and although they do share many characteristics, the molecular mechanisms associated with their pathology do in fact show differences. While the cause of CTE is ultimately injury-based, the cause of AD is unknown. I ultimately chose to compare these two unique tauopathies due to their extreme differences in both onset. Thus, deciphering their molecular similarities may provide a broad insight into potential therapeutic targets for tau-associated diseases. Thus, the goal of this presentation is to highlight the importance of tau pathology in AD and CTE for developing treatments through drawing connections between the diseases and their tau-related molecular mechanisms. Ultimately, the molecular characteristics of AD and chronic traumatic encephalopathy are very complex. Thus, this presentation intends to solely cover the prominent characteristics. Tau pathology in CTE is driven by repetitive head trauma, which causes mechanical stress and excitotoxicity. This leads to diffuse axonal injury, disrupting axonal transport and increasing intracellular calcium levels ultimately. The elevated calcium activates calpains, which ultimately cleave tau into fragments, which are more prone to aggregation and hyperphosphorylation. Unlike Alzheimer's disease, which we will discuss later, where amyloid beta triggers kinase activation, in CTE, mechanical stress activates kinases like GSK3 beta and CDK5, leading to NFT formation. Neuroinflammation and oxidative stress further exacerbate tau pathology in CTE. For example, microglia and astrocytes release pro-inflammatory cytokines, while oxidative stress damages mitochondria, which ultimately enhances tau hyperphosphorylation and aggregation. In Alzheimer's disease, GSK3 beta mostly drives tau pathology through excessively phosphorylating tau, again leading to neurofibrillary tangles. CDK5 also contributes by further phosphorylating tau and initiating aggregation. Again, this hyperphosphorylation ultimately impairs tau's ability to stabilize microtubules, which disrupts axonal transport. And this amyloid cascade hypothesis that I have here suggests that ultimately amyloid beta oligomers activate kinases like GSK3 beta, linking amyloid beta to tau hyperphosphorylation and neurodegeneration, which is ultimately the dominant difference between CTE and Alzheimer's disease. And further, chronic neuroinflammation through cytokines and on top of oxidative stress ultimately exacerbates tau pathology. These interconnected mechanisms highlight key therapeutic targets to ultimately interrupt the cycle of neurodegeneration in Alzheimer's disease. In both of these diseases, tau pathology is characterized by hyperphosphorylation, leading to microtubule destabilization and the formation of neurofibrillary tangles, or NFTs, which ultimately disrupt neuronal function. And that's sort of the main similarity between these two distinctive diseases. However, additionally, neuroinflammation, which is driven by activated microglia and astrocytes, exacerbate tau pathology in both diseases, which presents another common target for therapeutic intervention. Potential therapeutic targets for tau pathology in Alzheimer's disease and chronic traumatic encephalopathy include inhibiting kinases like GSK3 beta, like I was discussing before, and CDK5, which are dominant in those molecular signaling cascades for hyperphosphorylation, which we would need to monitor and inhibit in order to prevent tau hyperphosphorylation. We could do this by using small molecules and antibodies to ultimately block aggregation, which is the result of hyperphosphorylation. So that's definitely another target that addresses both diseases. And additionally, modulating neuroinflammation with anti-inflammatory or immune modulating therapies may also prove to slow disease progression. However, therapeutic targets in neurodegenerative diseases are highly under-researched. Thus, it's difficult to completely say that any of these therapeutic targets would actually be efficient to a broad range of tauopathies. Thus, this research ultimately helps to inform targeted therapies that can address the broad concerns over tauopathies without a specific diagnosis. Thus, it is possible that future research into the implementation of these therapeutic targets for neurodegenerative disease have the potential to be preventative among early onset. Thus, future research should further investigate these targets that I had identified as a result of those molecular mechanisms that are similar in Alzheimer's and chronic traumatic encephalopathy, and ultimately more research should be centered upon the molecular differences of tauopathies in order to create specified therapeutic targets. For example, we were talking about the amyloid cascade in Alzheimer's disease versus mechanical stress that triggers the hyperphosphorylation in chronic traumatic encephalopathy. These are the targets that should ultimately be addressed. However, this research aims to address the similarities and use those similarities to identify therapeutic targets, because this would address a broad range of tauopathies that have not had specific diagnoses. I would like to acknowledge my mentor, Nalini Rao. Thank you so much, and here are my references. Thank you.